Just want to go ahead and get started over here with our forward-looking statement. Many of you know what we do at Ocular Therapeutix, so I'm not going to spend a lot of time on that. We are developing drug delivery technologies for the front of the eye as well as for the back of the eye, and we also have a commercially approved sealant. Our vision is to transform ophthalmic care using sustained therapies. So last year when I stood up at this podium, we were talking about our upcoming milestones, and in glaucoma we were going to initiate a phase 2b trial. We have completed that study and have reported on those results. I'll talk briefly about that today. And on allergy, we were exploring our sustained release dexamethasone formulation OTX DP, which is now called Dextenza. And we actually finished that study and actually finished a phase 3 study also successfully and are reporting top-line results on that. On the post-operative front, we were planning to finish 2 of the phase 3 trials. Those trials were completed, and we successfully achieved the pain endpoint and filed our NDA. We are conducting a third phase 3 trial to pick up the inflammation indication, so that's going well also. On the anti-VEGF front, we were demonstrating feasibility of our anti-VEGF agents, and happy to show that those feasibility experiments have been quite successful, showing stability, release, and tolerability of these proteins. So a pretty full deck on that front. Just looking at implications of sustained release therapy, I think it's important to understand what role it plays in ophthalmology, and certainly is going to improve compliance in a huge way in diseases such as glaucoma, where less than 50% of patients are compliant over 6 months. But more importantly than that, there is compliance, but there's also the aspect of moving therapy from the hands of the patient to the hands of the physician, and thus assuring that complete therapy is given in a much more convenient form. And while we do this, we elute these drugs out at a much lower concentration, so they don't have the peak dose associated side effects. And since they're terminally sterilized, they are preservative-free, thus preserving the ocular health. So those are the implications of sustained release therapy. Now, when we are doing these types of studies, these are a little bit different than the classic phase 2 studies, for example, that biotech companies might conduct. We're using drugs that have a known mechanism of action and a known efficacy. So there is not that question whether the drug will have an effect or not. What we are instead trying to figure out is, because we give a one and done or once in several months type of a therapy, will there be enough dose to have the appropriate amount of effect? What is the level of effect? What is the duration of effect? What are the appropriate clinical endpoints, and what is the appropriate population? So we try to titrate those results in these phase 2 experiments, move to phase 3, and after that, once the phase 3s are successful, work towards indication broadening. So we're doing that with Dextenza, our sustained release dexamethasone product, where we demonstrated the absence of pain in a phase 2 study and in 2 phase 3 studies using this information and a great safety profile that we got with minimal or no late intraocular pressure spikes. We have submitted our NDA to the FDA, and we hope for an approval sometime next year. Expanding the indication into allergic conjunctivitis, we were glad to report on this phase 3 study where we met all the 3 parameters that the FDA guides towards, which is showing statistical improvement over placebo, showing at least half unit improvement at all time points, and at least a 1 unit improvement over majority of time points in ocular itching in a conjunctival allergen challenge model. So we achieved success in that, and we are working towards expanding Dextenza's label from pain, including inflammation and allergy, and we hope to begin the second phase 3 trial here shortly. So this video just shows how Dextenza is inserted. It's a brief process. It takes just a few seconds to do. The intracanaliculaar insert is placed over there. It can be visualized because it is fluorescent. So if you shine a light on it, you can actually, with a blue light and a yellow filter, actually visualize the placed product quite easily. So a patient can actually self-identify this. So moving from there onwards to OTX TP, our sustained release Travoprost formulation, we've conducted 2 phase 2 trials. Both showed a clinically meaningful level of efficacy of 5+ units. And when we looked at some of the results from the intent to treat as well as a post hoc analysis of the study we recently completed, we again saw that 5 units of improvement. The only surprise that we found was that timolol gave a performance a little larger than we expected. So the differential was about 1.9 units. We'd like to see it less than 1.5 units if possible. In the post hoc analysis, where we exclude patients on more than 1 drop, as well as looking at a later washout baseline, we find that that difference narrows. So we're going to take this information and some of our additional learnings and work with the FDA to come up with a phase 3 study design. Importantly, the phase 2A study results and B results showed that there were no serious adverse events. There was no hyperemia noted because we don't have the peak dose associated issues, and patients were able to visualize their depots quite accurately, and that concurred with what the physicians were seeing. And we were able to improve the retention of these depots, which is an important aspect also. So overall, pleased with this. What's in the pipeline? We're looking at dry eye as a potential indication. We have Dextenza in an exploratory phase 2 dry eye study, which will hopefully read out here in the next month or so. That's an exploratory study. But we are also looking at cyclosporin in preclinical models where we want to keep the levels of cyclosporin in between the 1 hour and the 12-hour levels and maintain it out to 3 months with a single punctum plug. So excited about the levels we're seeing in canines right now, and we hope to develop this further. On the anti-VEGF front, as we've mentioned in the past, we're working both on the protein delivery side as well as on a small molecule TKI side of things. The protein will be partnered. We are in late-stage discussions with several partners, and we hope to give more guidance towards that relatively soon. On the TKI front, for the first time, we're sharing some results over here where we, in a VEGF-induced leakage model, the picture on the left shows the leakage that takes place with the VEGF challenge, and by administering a single injection of TKI, which will elute out up to 6 months using our depots, we're able to block that leakage. This is a multifactorial. It inhibits VEGFR2 as well as PDGFR and TIE2, and has shown good tolerability. So we're excited about this development. So net-net, what do we expect to do this year? We want to report out on the phase 2 for exploratory study on dry eye, and we hope to begin our second clinical trial for allergic conjunctivitis so we can amend our NDA going forward, finalize the trial of the glaucoma study, gain our NDA approval for post-surgical pain, and then expand that NDA with the 2 supplements for allergic conjunctivitis and post-surgical inflammation. Finally, we hope to move further on the TKI front and continue that development for back of the eye diseases.